Welcome to this exploration of CRISPR, one of the most groundbreaking technologies in modern biotechnology. CRISPR is a revolutionary gene editing tool that allows scientists to make precise changes to DNA, the blueprint of life. This technology has transformed the field of genetics and opened up incredible possibilities for treating diseases, improving agriculture, and advancing scientific research. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. While this name sounds complex, it simply describes a special pattern found in bacterial DNA. These are repeating sequences of genetic code with unique spacers in between. Scientists discovered this pattern and learned how to harness it as a powerful tool for editing genes with unprecedented precision. CRISPR didn't start in a laboratory. It's actually a natural defense system found in bacteria and archaea, which are microscopic single-celled organisms. These tiny life forms use CRISPR to protect themselves against viral infections. When a virus attacks, bacteria capture pieces of the viral DNA and store them in their CRISPR sequences. This creates a molecular memory that helps them recognize and destroy the same virus if it attacks again. CRISPR technology relies on two essential components working together. First is the guide RNA, which acts like a molecular GPS system. It contains a sequence that matches the target DNA and guides the entire system to the exact location where editing is needed. Second is the Cas9 protein, which functions as molecular scissors. Once the guide RNA locates the target, Cas9 cuts both strands of the DNA at that precise location. The CRISPR process follows three main steps. First, the guide RNA finds and binds to the matching DNA sequence in the genome. Think of it as a search function that scans through billions of DNA letters to find the exact target. Second, the Cas9 protein cuts both strands of the DNA at that specific location, creating a break. Third, the cell's natural repair mechanisms kick in to fix the break, and this is where the actual editing happens. When DNA is cut, cells can repair it using two different pathways. The first is called non-homologous end joining, or NHEJ. This pathway simply joins the broken ends back together, often introducing small errors that disrupt the gene's function. This is useful when scientists want to turn off a gene. The second pathway is homology-directed repair, or HDR. This is more precise and allows scientists to insert new genetic code by providing a template for the cell to copy during repair. CRISPR has enormous potential in medicine. Scientists are using it to treat genetic disorders like sickle cell anemia, where a single genetic mutation causes red blood cells to become misshapen. By correcting this mutation, CRISPR can potentially cure the disease. Researchers are also developing CRISPR-based therapies for cancer treatment, using it to enhance immune cells so they can better recognize and destroy cancer cells. Beyond human health, CRISPR is being used to create disease-resistant crops that can help address food security challenges. CRISPR has several advantages that make it superior to older gene editing methods. First is precision. It can target specific DNA sequences with remarkable accuracy among the 3 billion letters in the human genome. Second, it's cost-effective. CRISPR is significantly cheaper than previous technologies, making genetic research accessible to more laboratories worldwide. Third is speed. The editing process is much faster than traditional methods, accelerating research timelines. Finally, CRISPR is incredibly versatile, working across different organisms from bacteria to plants to animals, including humans. Despite its promise, CRISPR faces several important challenges. Off-target effects are a major concern. Sometimes the system cuts DNA at unintended locations that are similar to the target sequence, potentially causing harmful mutations. Delivery methods also pose challenges. Getting CRISPR components into specific cells and tissues in the body, especially in living organisms, remains technically difficult. Additionally, there are significant ethical concerns, particularly around editing human embryos or germline cells, where changes would be passed on to future generations. CRISPR represents a revolution in biotechnology, offering unprecedented precision and accessibility in gene editing. As research continues, scientists are developing improved versions with better accuracy and fewer off-target effects. The technology is expanding beyond simple gene disruption to include base editing and prime editing, which allow even more precise changes. CRISPR is transforming our ability to understand genetics, treat diseases, and shape the biological world around us. The future holds immense potential as we continue to refine and responsibly apply this remarkable tool.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.